Okay, we are on page 111 in your notes, page 111. The reason why I kind of jump around like this, there is a method to it. I don't like to go in order the how the textbook goes, but I like to kind of build up and just make it make more sense. To me, this makes more sense to go in this direction. And so our next topics are mitosis, cancer, embryo development, and stem cells. And the reason why I picked that as our next topic is because we just talked about gene regulation. And gene regulation is going to be very, very important in cell division and embryo development. So the cell cycle, that's the first blank you'll see on page 111, is the life of a cell from its formation until it divides. So the entire life of a cell. Believe it or not all cells are always dividing. The functions of cell division for cells who are needing to divide are for reproduction, growth, and tissue repair. And this is just showing you some pictures, and I know on your paper they look kind of ugly, but this is just showing you pictures of cell division, growth and development, tissue renewal, and various types of cells. Just some vocab words that we need to go over real quick. Genome. Genome is all of the cell's genetic info. DNA, remember before, why do I say this word? Genome, before a cell divides, all of the DNA has to replicate and copy before the cell divides. So the genome has to replicate before a cell divides. Remember, a prokaryote has a single circular chromosome. A eukaryote has more than one linear chromosome. And just so you know, I want to start talking a little bit more about chromosomes, especially when we get into genetics later. I need you to know that humans have 46 chromosomes. 46. 23 sets of 2, one from each parent. So 23 types of chromosomes, one from each parent. That equals 46. A mouse has 40, and a fruit fly has 8 chromosomes. And what this is showing you is a cell, and this is the nucleus, and that's the condensed chromatin within the nucleus of a cell that's probably getting ready to divide. Prokaryotes are going to divide by a process called binary fission. So on your paper, it says bacteria cells divide by binary fission. That's the same thing as prokaryotes. Eukaryotes divide by a process called mitosis. So we always talk about the differences between prokaryotes and eukaryotes. Here's one big difference. Prokaryotes divide by binary fission. Eukaryotes divide by mitosis. And so the topic of this lecture is mitosis. But before I move to mitosis, I'm going to talk a little bit about prokaryotes and what binary fission is. And so if you look at that picture, you'll see you have a cell, a bacterial cell. Step 1, chromosomes replicate, DNA replication. Step 2, replication continues. Step 3, replication finishes, and the cell splits into 2 daughter cells. Really simple. So binary fission is the way that bacterial cells copy, replicate DNA, and then they split in half. That's how they reproduce. That is how they replicate, basically. With eukaryotes, just like anything else, eukaryotes are going to be a lot more complicated and are going to divide by a process called mitosis. So each chromosome must be duplicated before the cell divides. That's replication. We already went through that. A duplicated chromosome equals 2 sister chromatids attached by a centromere. So pay close attention here, because this is where students get really confused. We have a lot of C words like chromosome, chromatid, centromere, centriole, centrosome. You're going to need to know the difference between all of those words by the end of this lecture. And so the first C word is chromosome. Chromosome is just condensed chromatid. Chromatin is just condensed chromatin. When a chromosome is copied, it is called 2 sister chromatids. So if you look here on step 1, there's just 1 chromosome. Step 2, that chromosome has duplicated and they're 2 sister chromatids. That's why chromosomes are always represented by an X, because it's 2 copies. And then in the very middle, they're connected by this thing called a centromere. So I would go ahead and start making flashcards for all of this. Then when the cell divides, each sister chromatid gets separated from the other and goes into the new cell. This is under an electron microscope. And sorry, I have an air conditioner going on behind me. This is under an electron microscope. This is what two sister chromatids looks like in a centromere. So remember, it's one chromosome with its copy attached by a centromere. Those two get separated during mitosis and each go into their own cell. All right, so a little bit more vocab to go over is somatic cells versus gametes. And the reason why I'm kind of just moving quickly through this because this is a review from Cell Bio. There really shouldn't be anything new except for when we talk about cell cycle regulation. There shouldn't be anything new though here. Somatic cells are also known as body cells, like your skin cells, your liver cells, your stomach cells, your eyeball cells. They're diploid or diploid, meaning they have two of each type of chromosome. So in a human, a diploid cell, a somatic cell, would have 46 chromosomes, 23 pairs, 46 chromosomes, one from mom, one from dad. Somatic cells will divide by mitosis, and like I had said, a human is diploid and it has 46. Don't you like how fill in the blanky this is? Ugh, I was so nice on this lecture. There's not a whole lot to write. You're just filling in some blanks. But that's great because we can go a little bit quicker whenever they're like this. I want to see the notes like from here on out are a little bit more fill in the blanky. All right, so those are somatic cells, body cells, diploid, divide by mitosis. Gametes are going to be sperm and egg. So if it is not a sperm or an egg, it is a somatic cell. If it is a sperm or an egg, it is a gamete. They are considered haploid. They only have one of each type of chromosome. And that should make sense because one sperm and one egg fuse together to make a diploid cell. So you have two haploids that come together to make a diploid, and then that divides by mitosis to make an actual organism. So you need to know the word somatic, the word gamete, diploid, haploid, mitosis, and then meiosis. Gametes divide by meiosis. And in humans, a human gamete, a human sperm, or a human egg are only going to have 23 chromosomes. All right. Pause if you need to, because I'm going to move on. Let's talk a little bit about the cell cycle. The cell cycle is the entire length of a cell's life. Okay, we start right here where this arrow is, and we move clockwise, 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 all the way to mitosis, and then it starts over right here. So what I want you to see is that the order is G1, S, G2, M. G1S, G2M. Then this cell will undergo mitosis and then cytokinesis. So that's the order. So I need you to know the order of the cell cycle steps. G1S, G2M, cytokinesis. All right? The mitotic phase is going to alternate with interphase. So I'm going to go back to the picture. Yellow is mitosis. Everything else is interphase. So if it's in G1, S, or G2, it's in what we call interphase. That means the cell is not dividing. So if it's in G1, S, or G2, it's in interphase. If it's dividing, it's in mitosis. All right? And then that's the order they go in, so I already said that. Interphase, 90% of the cell cycle, 90% of the time the cell is going to be in interphase. Interphase is going to include G1, which is where cells grow and carry out normal functions, S, which is when chromosomes duplicate. So what I want you to write next to S is DNA replication. So G1 is growth, S is DNA replication. The S stands for synthesis. So it's the synthesis of DNA, DNA replication. And then G2, the cell is growing more and preparing for cell division, right? So pause if you need to. So if I asked you which part of the cell cycle is the cell in for the most amount of time, you would say interphase. And then if I asked you what are the three parts of interphase, you would say G1, S, and G2. All right, mitosis is not part of interphase. Mitosis is where the nucleus divides and then the cytoplasm divides, and then the cell becomes two different cells. So that is all I'm going to say about G1, S, and G2, because we've already talked about S phase. That was chapter 13, DNA replication. I'm going to talk about all of the steps of mitosis now and cytokinesis. This should look very familiar to you. The picture on your notes turned out super dark and ugly. Always refer to your textbook. This is chapter 9. So if you look in your textbook, this is chapter 9. Mitosis has very specific steps. The steps are prophase, metaphase, anaphase, telophase. So the way you can think about it is PMAT. So there's the 4 steps of mitosis: prophase, metaphase, anaphase, telophase. And then you have pictures here. The pictures on top are real pictures taken with an electron microscope or a fluorescent microscope. The ones on the bottom are just drawings or models. And so mitosis is a continuous process with observable structural features. The chromosomes condense and become visible during prophase. So you're going to write down prophase. And then you have a picture down here to refer to. The chromosomes are condensing and becoming more visible. I'll never ask you a question about prometaphase. Prometaphase is just the in between prophase and metaphase. So you can just scratch that out. It's not a big deal to know that. Chromosomes line up at the equator. Equator means middle. So that's what helps me remember metaphase. It starts with an M, and so does the word middle. Chromosomes line up in the middle, and you can see that here in this picture. So you have these spindles that form. Remember, spindles are made up of microtubules, right? And the spindles grow out of the centrioles. Okay, so that's back from chapter 4. All right, metaphase, the chromosomes line up in the middle. They're on those spindles, which are made up of protein microtubules. All right, anaphase is the separation of sister chromatids. Anaphase starts with an A. The word apart starts with an A. That's how I remember anaphase. Sister chromatids, which is a chromosome and its copy, actually get pulled apart. And then telophase and cytokinesis is where the nucleus divides and then the cytoplasm divides. And I'm sorry, there's people behind me talking, but I'm sure you can still hear me. All right, I would definitely refer back in the book. Just reference these pictures. Or you can get this off of Veracross. This slideshow is on Veracross. All right, so you need to know the PMAT, prophase, chromosomes are condensing, metaphase, chromosomes are lining up in the middle, anaphase, chromatids are getting pulled apart, telophase and cytokinesis is the division of the nucleus and the cytoplasm. All right, and then this is just showing you the pictures again. Prophase, so what do I want you to know? Prophase, metaphase, anaphase, cytokinesis, and telophase. So let's look a little bit at cytokinesis. So cytokinesis is the cytoplasm dividing. And so in animal cells, it's going to form this thing called a cleavage furrow. So you're going to have these, it's almost like rubber bands wrapping around that pull tighter and tighter until the cell like splits. In plant cells, you're going to have a cell wall forming. So you're going to write that down. In animal cells, you have something called a cleavage furrow. And then in plant cells, you have a cell plate that forms. And you have some pictures there. So with animal cells, you're going to have what's called a contractile ring of microfilaments, kind of like a rubber band that's squeezing tighter and tighter until it splits. And then in plant cells, you're going to have a plate that forms in the middle. And this is showing you, so here's the contractile ring of microfilaments right here. It pulls and pulls until it eventually splits. With plant cells, because of that cell wall, it can't do this. It can't wrap something around and like split it. So what you have is one big rectangle and then a plate that forms in the middle. And that is how cytokinesis differs between animal and plant cells. So you're going to need to know how cytokinesis differs between the two. You're going to definitely need to reference your book. With this picture right here, this is just showing you what it would look like under a microscope. I would be familiar with these pictures, so make sure you go to chapter 9. You guys are fully capable of turning the pages and looking at the pictures, and you're supposed to be reading the textbook anyways to help you out. So I would definitely read the textbook and definitely reference these pictures. What I would actually do on page 115, I would redraw the pictures since they're not showing up. So prophase chromatin is condensing, metaphase, they're lining up on the metaphase plate, anaphase, they're being pulled apart, and then telophase and cytokinesis is the nucleus and cytoplasm dividing. A plant cell is usually our go-to because it's easier to see them under a microscope. We might attempt to do this in class, but again, prophase chromosomes are condensing, metaphase, they're lining up in the middle, anaphase, they're being pulled apart, telophase and cytokinesis, the nucleus and the cytoplasm are dividing. I would definitely be familiar with these pictures. And so we're going to do an activity in class where we identify the phases of mitosis. So we're not going to do that today. Then the last thing is that during anaphase, so remember anaphase is the phase where chromatids, sister chromatids are being pulled apart. What I need you to know is how they are being pulled apart. So you have your chromatids that are attached to the spindle, the microtubule spindle, with a protein called a kinetochore. So you need to know that word kinetochore. So you have a protein called a kinetochore that's attached to this, that attaches the chromatid to the microtubule spindle. The kinetochore then chops the spindle as it goes. So it almost is like a walking motion. So chromosomes walked to the poles by motor proteins. So they like walk down the microtubule spindles with a motor protein that is inside of this protein called a kinetochore. All right, so the kinetochore microtubules shorten at the end as they depolymerize. That just means that the spindle is getting chopped as it goes. So if you can imagine the chromatid moving down the spindle and the spindle getting, it's walking and it's chopping at the same time. So walking and chopping at the same time is kind of how I can explain that. And then the kind of greenish teal looking thing is the kinetochore. That's what attaches the chromatid to the spindle. And then the red thing is the motor protein that walks it down the spindle. And then what this is showing you, the tubulin subunits are the chopped up spindle. As it walks, it chops it up. All right. So make sure you let me know at the end if you have questions on that. Okay, we need to talk about checkpoints and the regulation of the cell cycle. Regulation of the cell cycle, I'm on page 117. So during the whole cell cycle, not just mitosis, now I'm going back to interphase, which includes G1, SG2, and mitosis. There are checkpoints where there are stop and go signals that regulate the cell cycle. All right. So you're going to want to write that down. There are checkpoints that are control points where there are stop and go signals that regulate the cell cycle. It's very important that these checkpoints work so that a cell doesn't underdivide, but also so it doesn't overdivide. All right. So pause if you need to keep writing because I'm going to move on. The G1 checkpoint is the most important. So we look back at the picture. G1 checkpoint is right here. Conveniently, it's within G1. The G1 checkpoint is the most important. It is controlled by cell size, growth factors, and the environment. So if the environment isn't conducive to cell division, it won't happen. If the cell's not big enough, if there's not growth factors present, the cell won't go forward. If a cell passes the G1 checkpoint, it will be committed to completing the whole cell cycle. So what the G1 checkpoint does is it commits the cell to division. You need to write that down. The G1 checkpoint commits the cell to division. If a cell does not pass the G1 checkpoint, it enters in this thing called a non-dividing state, also known as a G0 phase. Nerve cells, muscle cells, and liver cells will all go into a non-dividing state. Nerve cells and muscle cells will stay in a non-dividing state. Liver cells can be called back from G0. So they're going to be non-dividing unless something happens, something triggers them to divide. Okay? So that's the G1 checkpoint. Most important, You need to know what it's controlled by. You need to know that the G1 checkpoint, if it is passed, it commits the cell to division. You need to know that if it doesn't pass the G1 checkpoint, then the cell is going to enter this G0 non-dividing state. And you need to know that nerve cells, muscle cells are always in a non-dividing state. Liver cells go back and forth. The G2 checkpoint, so let's look back at the picture where that is. It's right here. So G1 checkpoint puts it into DNA replication. So S phase occurs, which is DNA replication. I ask questions about that, so you're going to need to know that S phase is DNA replication. And then it goes through the G2 checkpoint right before it goes through the G2 phase. Right before mitosis is when it needs to pass the G2 checkpoint. So write that down, right before mitosis. So G2 checkpoint is controlled by DNA replication, DNA mutations, and cell size. What it's doing is it's checking to make sure DNA replication occurred correctly. Okay, so I would write that down. The cell's not going to enter mitosis unless DNA has replicated correctly, and then refer back to the picture. Well, that G2 checkpoint is right before mitosis starts. If DNA has not replicated correctly, that cell is going to undergo apoptosis. It's not going to divide. All right, and then you have the M checkpoint. Notice in the picture at the top of page 117, or I'm going to actually go back to it. Notice where the M checkpoint is. It's right in the middle of mitosis. So what the M checkpoint is doing is making sure that the spindle has formed correctly. And you guys know from cell bio that you have the centriole. The microtubules grow out of the centriole to form the spindle. The spindle is what the chromatids attach to and what aids in them pulling apart. Okay, so the M spindle metaphase checkpoint is in the very middle of mitosis. It checks to make sure the spindle is formed correctly and that chromosomes are attached to their kinetochores correctly. All right, this is just a picture showing you if a cell passes a G1 checkpoint, it's committing to dividing. This is showing you if a cell does not pass the G1 checkpoint, it goes into a non-dividing state like nerve and muscle. This is showing you right here on number 3, the M spindle checkpoint, making sure that the chromatids and their kinetochores are attached to the spindles correctly. So really all of this picture is showing you is again, what I showed you a minute ago, kinetochores are little proteins on the chromosomes that attach them to the spindles. Make sure that they're attached correctly. Because if they're not attached correctly, you could end up with a cell that has an extra chromosome. And we're going to talk all about the disorders that can result from that. All right. So those are where the cell cycle checkpoints, the 3 checkpoints. So you need to know those 3 checkpoints. Let's talk a little bit about those checkpoints. So you see up here G1S, G2M, G1S, G2M, and then you have these chemicals and it's showing you how they fluctuate. So you have this orange curve and then you have the purple line, basically. And so what I need you to know is that MPF is called the maturation promoting factor. Yeah, maturation promoting factor. We'll get to that in just a second. So there's chemicals that fluctuate during the cell cycle that help push a cell into mitosis. And we're going to talk about what are these compounds, what are these chemicals that help push a cell into mitosis. So you have these things called kinases. You've seen the word kinase before. They're enzymes that control the cell cycle. So you have kinases, and then you have other proteins called cyclins. So what do I want you to know? What I want you to know is that cyclins are going to cyclins, yes, are going to activate kinases. So cyclins are going to activate kinases. And you see how the cyclin levels go up during mitosis, okay? So cyclin levels go up during mitosis. And so then I ask the question, then you see this MPF, this is called maturation promoting factor. The maturation promoting factor is a cyclin bonded to a kinase. So I should have showed you that first. So maturation promoting factor is a cyclin and a kinase bonded together that allows a cell to pass from G2 and go to the M phase. So just kind of write that down, and then I'm going to go back and kind of show you what we're getting at here, because this can be very, very confusing. And then we're going to end on page 119. So this is a really, really confusing thing. I wouldn't just gloss over it. I would go back and make sure you understand it. All right. So these are internal regulatory molecules. So let's just kind of go back. So pause if you need to write, and then push play again, because I want to go back and re-explain this. All right. So in order to push a cell past the G2 checkpoint, you have these two proteins that are controlling it. They're called kinases and cyclins, okay? That's what I need you to know from this slide. In order to push a cell past the G2 checkpoint into mitosis, you have two proteins that are controlling that. They're called kinases and cyclins. When a cyclin and a kinase, so a kinase is also called a CDK, cyclin-dependent kinase, because when this level goes up, the cyclin is going to control the kinase, basically. So what this is saying here is kinases are always there. You can see the CDKs are always there. When cyclin levels go up, it binds with the kinase, and it's the binding of the cyclin with the kinase that pushes the cell past the G2 checkpoint. I feel like this could have been arranged in a much different way to be less complicated. So maturation promoting factor is what pushes a cell past the G2 checkpoint. It involves a cyclin binding to a kinase. The cyclin levels are going to control this whole thing because kinases are always there. I'm probably going to have to re-explain that during class, though. Just try to understand it. All right? So these are just proteins that are going to push a cell past the G2 checkpoint. They have to be very, very specific levels in order to work. All right, so those are internal. And this can get a lot more complicated. I'm just showing you guys some images of how complicated it actually gets, and that there's a lot of proteins that are involved in controlling the cell cycle. That is just an example of one. A cyclin binds with a kinase, forms a maturation promoting factor, pushes the cell past the G2 checkpoint. All right, that's internal regulatory factors. It's an example of an internal regulatory factor. Next, we have external regulatory factors. And we have three: anchorage dependence, density dependence, actually just two: anchorage dependence and density dependent. So growth factor are proteins released by other cells that stimulate cell division. So that's going to cause a cell to divide. Okay. Then we have density dependence, and then we have anchorage dependence. So this is just showing you the three things. So there's three external regulatory factors. Growth factor, which is a signaling molecule released by other cells that cause the cell to divide. And then you have what's called density dependent inhibition. Crowded cells normally stop dividing. So it's called density dependent. So when cells get crowded enough, a signal is sent to stop dividing. And then we have anchorage dependence. Cells must be attached to another cell or an extracellular matrix in order to divide. All right? So all of these things that we talked about. Cyclins and kinases can be disrupted. That can lead to cancer. Growth factor receptors, remember we talked about receptor tyrosine kinases and growth factor signals. That can be disrupted, and that can lead to cancer. Cancer cells are going to lose density dependence. They're not going to be able to sense when there's too many cells around them and to stop dividing. So they're going to lose that. And then cancer cells eventually lose anchorage dependence. They will keep dividing even if they're not attached to something else. And so our next topic is going to be cancer and the genes that are involved in cancer. All right, and so I'm just going to stop there, and we will do cancer in class next time I see you. Bye.